Good day, everyone. We're reviewing Wetzel's Limnology textbook, and this is edition number 4, and we are on chapter 9, which is structure and productivity of aquatic ecosystem. And today we'll be looking at metabolism. So let's jump into that chapter. Ecosystem metabolism describes the turnover of biomass and energy. Measuring all the inputs and outputs of energy and biomass can be a very time-consuming way to calculate net ecosystem production, or NEP. NEP is the difference between GPP, which is the gross primary production, and respiration. With the increased availability of high-frequency dissolved oxygen probes, the diel oxygen method has become widely used to determine the ecosystem metabolism of inland waters. To facilitate comparison and use of different metabolism methods, Winslow developed a software package that implements different models that estimate lake metabolism based on in situ high-frequency time series of dissolved oxygen and temperature. As with other methods that quantify aquatic metabolisms, this method comes with its own set of limitations and assumptions. For instance, an underlying assumption of the diel oxygen method is that respiration measured during night times resembles respiration during daylight, which may lead to a significant underestimation of respiration during daylight. In addition, the uncertainties associated with this method decrease considerably when precise measurements of wind speed at the water surface are used as inputs, which is rarely done in practice. Aquatic food webs. In self-sustaining biological communities, functionally similar organisms can be grouped into a series of operational levels of producers and consumers, together constituting a food web. The trophic structure of a community refers to the pathways by which energy is transferred and nutrients are cycled through the community trophic levels. The basis of the aquatic food web is formed by primary producers that are able to fix carbon by photosynthesis. Primary producers in aquatic food webs include phytoplanktonic algae and photosynthetic bacteria, benthic algae and photosynthetic bacteria, and aquatic plants. Primary producers are eaten by primary consumers, herbivores or omnivores, which are in turn eaten by secondary consumers such as carnivores and omnivores. Consumers in aquatic food webs include zooplankton, benthic animals, fish, terrestrial animals, and estuarine, marine animals. Often neglected in studying aquatic food webs, parasitism is arguably the most commonly occurring consumer lifestyle. Autochthonous plant material not consumed until after it dies joins the pool of detritus, which also includes allochthonous dead plant material, dead animals and feces, consumed by omnivores and detritivores. Omnivory is prevalent in ecosystems. Few species feed entirely on either live plants, live animals, or detritus. In most ecological communities, individuals of a species feed from several trophic levels. Furthermore, many animals that feed on live or dead plant material are actually gaining much of their nutrition from insects or microbes living on the plant material. The use of stable isotopes, together with feeding observations and gut contents, has enabled ecologists to separate what an organism consumes from what it assimilates into its tissues. This has confirmed that omnivory is prevalent and that species differ in their capacity to change their diets in response to food availability or competition. Indeed, it is now perhaps better to divide species into flexible or inflexible consumers than into herbivores, omnivores, detritivores, carnivores, or scavengers. Omnivory also occurs across life histories as a result of individuals using different resources during different parts of their life cycles. Many of these differences in feeding, particularly during the different life stages and sizes of the species, will be detailed in the discussion that follow in this work. The amount of energy available for metabolism decreases with each increase in trophic level because energy is lost with each transformation. Since organisms expand a considerable amount of energy for maintenance, only a portion of the energy of one trophic level is available for transfer and used by higher trophic levels. Total biomass and numbers of individuals at each successively higher trophic level also usually decrease, although some exception will be discussed later. In addition to flows of energy, elements such as phosphorus and nitrogen are also transferred through the food web. Ecological stoichiometry provides a compelling framework to understand the effect of the imbalance, or the balance, of such chemical elements on ecological interactions. Stoichiometric constraints may arise when, through human-induced increase in atmospheric nitrogen deposition or climate warming, the changes in the chemical balance of the primary food web results in lower food quality for higher trophic levels. 9. Population structure, growth, and regulation. A population comprises a subgroup of a single species that are interbreeding. Ecologists use molecular genetics to define the spatial and temporal extent of aquatic populations. The number of individuals per unit area or volume of water, the population density, is an index of population size at any given time. Ecologists sometimes assume that the representative of a species at a particular location are in different population from those at other locations. This assumption may appear reasonable when comparing a number of lakes, but is less acceptable when comparing sites in the same river catchment, which may be connected by flow. However, molecular genetics has shown clearly that any sample of invertebrate species from a single site will contain some species that do make up a single population at a site, and others where a single population covers a much wider area. For vertebrates, for example, fish, water birds, a single population may occupy an area of hundreds of square kilometers. The area that a population occupies is determined by 3 variables: the dispersal ability of the species, the distribution of suitable habitats within the surrounding area, and the connectivity permitted by the landscape. For freshwater species, landscape connectivity may mean connection via surface flow or underground water movement, ribbons of fringing vegetation along rivers, or overland via flights. Unfortunately, human activity in landscapes has often altered both connectivity and the distribution of suitable habitats, and in doing so has altered the distribution and population size of microbes, plants, and animals in freshwaters. So this is figure 9-7. Common sigmoid growth curve of a population with time nt as it approaches the environmental carrying capacity k. The growth rate, the change of n over time, denotes the difference in the number of organisms over each time interval. Okay. So we have a population number here, and so on the y-axis, and here we have, so this curve is growth rates. So this is the change of the population over time, and I'm assuming here we are on the x-axis, we're dealing with time. So there's a population growth, and you can see that the ecosystem has a carrying capacity for that population at that level. So it could sustain that level of population. However, you can see that the growth of the population also has an inflection point, start to decrease, because the population will be reaching the carrying capacity of the system. The growth of a population can be very rapid amongst smaller organisms, but restrictions imposed by the environment, predation, and intra and interspecific competition quickly limit the size of populations, either by increasing the death rate or by decreasing the birth rate. Populations have an intrinsic growth rate, that is how quickly they can reproduce under ideal conditions, but mostly there is a range of limitation on population growth. Density-dependent limitations are imposed by competition with others in the population for available environmental resources, by the production of waste products, and by behavioral factors. As the population increases toward the carrying capacity, maximum number that can be supported by available resources, fewer resources are available for reproduction, so reproductive rates decline. This produces a logistic growth curve, which has a sigmoid shape and has been commonly observed among natural and experimental populations. However, it is important to note that species may cheat on this equation, which are a simplification of reality. They cheat by using by use of trade-off. That is, they trade off body condition to achieve reproductive success. For example, a population of a species in a location where resources are scarce may reproduce at a smaller body size or younger age than under good conditions, and they may produce fewer or smaller offsprings. Those offsprings may start life at a disadvantage in size, numbers, or body conditions, but may be able to make up for that disadvantage if they experience better conditions for growth and development than their parents. When sampling a population, it may be possible to identify a large group of individuals of the same or nearly the same age or size, referred to as a cohort. Cohorts may be followed through time to estimate survivorship and understand a species' phenology. Phenology is the time at which important life cycle events occur. However, it can be very difficult to identify a cohort in a species that reproduces continuously, and as do many populations of freshwater plants and animals. Also, many freshwater animals have complex life cycles, where the juveniles rely on quite different resources and may even live in quite a different habitat to the adults. For example, many juvenile amphibians rely on the presence and duration of water and aquatic food, but as adults, they obtain most of their resources from the terrestrial environment, only returning to water to breed. Modeling such complexity is challenging, as the different life stages are exposed to different predators, environmental risk, and intra- and interspecific competition. For this reason, the use of structured population analysis, such as life tables and individual-based modeling approaches, have waned in popularity among freshwater scientists. Time series analysis have shown that the phenology of lake processes may be impacted by climate change, which could interrupt the trophic transfer of energy when producers and consumers are affected differently. Metapopulations. Metapopulation theory acknowledges that individuals within species are not necessarily sedentary, but may disperse among populations to interbreed. This interbreeding is essential to maintain genetic diversity within species. Essentially, a metapopulation is a group of patches occupied by subpopulations and connected by the dispersal of individuals among the patches where interbreeding occurs. Most populations of freshwater species are likely to be part of a metapopulation. Freshwater ecosystems are dynamic, and disturbances may extirpate a species from a water body. In a metapopulation, individuals will arrive back at that patch and reestablish the species there. So metapopulation also allow freshwater species to survive in spatially and temporally dynamic ecosystems. In a metapopulation, patches of habitat may differ in their function. Some patches may be sources, some may be sinks, and others may be stepping stones. That is, a species may be present in some patches even though it cannot complete its life cycle or breed in that patch. In source patches, the birth rate exceeds the death rate, and the emigration rate is therefore high. Stepping stone patches enable individuals to move longer distances across landscapes by providing sufficient resources to enable species to survive there briefly. Stepping stones patches have no birth rate, a low death rate, and high emigration and immigration rates. Sink patches also have no birth rate, and they have a death rate equal to the number of immigrants. Sink patches or ecological traps may or may not provide resources to species, but they are insufficient to support successful reproduction or recruitment. Understanding metapopulation dynamic is important for species conservation. For example, conservation reserves should aim to include source patches and be linked by stepping stone if needed. A conservation reserve that contains only sink patches for a species will not support that species for the future, even though the species has been recorded there. As climate change continues to alter water regimes and therefore the availability and connectivity of freshwater habitat in landscape, it will alter metapopulation dynamics and the relative frequency of source, stepping stone, and sink patches in landscapes. Communities. Although variously defined, a community usually refers to a group of interacting populations of different species within a defined spatial area. Some practical spatial boundaries are most often applied to studies of communities because habitats are diverse and often organisms move from one habitat to another. Communities are generally defined by a researcher interested in a particular group of species, usually occupying a defined environment. Sometimes the term is used erroneously, such as when it is used to describe a single taxonomic group, like for example the Trichopteran community. The term community should only be used where there are a variety of different taxa. Where only one taxon is described or one group of taxa, the term assemblage should be used. Community ecologists focus on the patterns and processes in the living parts of the ecosystem. They are interested in biodiversity and the processes that sustain it. The effect of disturbances on communities and pathways for recovery from disturbances. They are interested in interaction between species such as predation, competition, mutualism, and parasitism. They also seek explanation for the patterns of multi-species distribution in different places and at different times. These questions are important for managing ecosystems to sustain biodiversity and the natural processes that support it, and to help ecosystems be resilient to environmental change. The rapid progress of global warming and other global changes has increased efforts to understand community function, respond to negative impacts, and use restoration to try to sustain the Earth's remarkable biodiversity. Freshwaters are under exceptional pressure because of their importance to humans for water supply, power generation, transport, and waste disposal, and their vulnerability to climate change while also occupying a disproportionately small area of the Earth. Ensuring healthy, functioning freshwater ecosystem for the future is the main aim of community ecology and in freshwaters. Community assembly. There is an enormous amount of literature covering the debate about how ecological communities are structured. This debate has changed over the decades, but there is still vigorous discussion. Here we provide only a beginner's guide to some of these ideas, noting that there will be further development over the lifetime of this book. Why do species occur where they do? How are communities assembled? These are two of the most enduring questions in ecology. A range of factors are known to determine assemblage composition, and their relative importance varies from place to place and between times in a single location. These factors include environmental conditions, species traits, interactions between species, dispersal, and connectivity of landscape. And connectivity. Density dependence, as mentioned above, was one of the first concepts regarding limits of population. It includes both threshold limitations, such as the availability of territories or nest sites, and continuous limitations, such as food supply, the effect of which intensifies as population size increases. One aspect of density-dependent controls on population is that they may only be observable at particular times. Ecologists may observe only the after effect of a density-dependent variable without being aware that it has occurred. In another theory, Southwood defined the habitat template as the habitat provides the template on which evolution forges characteristic life history strategies. This theory is based on the premise that habitat conditions select for certain traits that reflect adaptation for survival and reproduction. This idea was most often applied by ecologists studying disturbances, which are natural events such as floods and droughts, or anthropogenic events such as pesticide influx into freshwaters. However, unless an ecosystem is being disturbed very frequently, this theory does not represent all the dynamics we observe in freshwater ecosystems. More recently, complex systems theory has been applied to freshwater ecosystems. Indeed, many of the studies that led to the development of this theory occurred in lakes. Complex systems display fluctuating nonlinear dynamics, so short-term phenomena may be hard to predict, but they are more predictable in the long term because they operate within particular boundaries. Indeed, because freshwater ecosystem provides so many ecosystem services, they are understood to be embedded in complex socio-ecological systems. Resilience is the ability of a system to recover following disturbance. It is a key element of complex systems. Therefore, managing resilience requires a thorough understanding of the functioning of both the social and the aquatic system, plus the interdependencies between them. The alternative stable ecosystem states observed in shallow lakes are an extreme form of nonlinear dynamics and will be elaborated in chapter 26.